clinical trial involving hundreds or thousands of patients can produce mountains of data pertaining to patient outcomes. In order to cut through the clutter, we use what's called a Kaplan-Meier graph. I'd like to invite you to spend the next few minutes with me as I explain what a Kaplan-Meier graph is and show you how to read one. Then you'll be able to see for yourself why it is that hundreds of randomized clinical trials aimed at identifying one-size-fits-all chemotherapy treatments have failed to improve cancer survival rates. The clearest way to present data obtained in a clinical trial is by using what is called a Kaplan-Meier graph. The purpose of a Kaplan-Meier graph is to make it easy to show at a glance when a specific event has occurred among patients enrolled in the clinical trial. The event which is depicted in a Kaplan-Meier graph is called an endpoint of the clinical trial. The three most commonly studied clinical endpoints are these: tumor response, progression-free survival, and overall survival. In our discussions here, the endpoint we will be considering is the one that matters the most: overall survival. In its simplest form, a Kaplan-Meier graph consists of two axes and two lines, or curves. Typically, each curve on the Kaplan-Meier graph represents a different group of patients. These groups are called cohorts. Patients are grouped together into cohorts based upon certain similarities which are shared by all patients in the group. In the studies we will be showing here and in upcoming modules, patients are grouped based upon the type of cancer they have, and also based upon the specific chemotherapy they have received in a clinical trial. Generally, one chemotherapy treatment will be compared to another chemotherapy treatment. The object is to see which treatment worked best for patients who have the same type of cancer. Let's see how the Kaplan-Meier graph works. The x-axis is the horizontal axis. It represents time. In a cancer clinical trial, time might be presented in weeks, months, or years. The y-axis represents numbers of patients. This value can be expressed as an absolute number of patients or as a percentage of patients. In virtually all clinical trials of new treatments, patients are randomized. That is, they are chosen at random to receive one therapy or another. I would much rather see each patient receive a treatment which was chosen for him or her through personalized cytometric profiling, as I've advocated for many years. Unfortunately, it isn't done that way in most clinical trials today. Getting back to our explanation, for illustration, let's call this curve Cohort A. It depicts patients who have received treatment A. Let's also say that 100 patients received treatment A. This makes it easier because it means that the number of patients and percentage of patients will be identical. In real life, it is seldom this simple. At the start of the study, all of the patients are alive. Each time a patient dies, the line takes a tick downward to indicate that the number of patients who are still living has decreased. As we move out over time, we can see that fewer patients are still alive. Finally, by the end of the study period, we can see that of the 100 patients who received treatment A, 75 patients, or 75% of the original cohort, are still alive. Now, in our hypothetical example, let's look at the second curve. We'll call this group of patients Cohort B and say that all patients in this group received treatment B. Let's say, again for the sake of clarity, that 100 patients received treatment B. Again, all patients in this cohort are alive at the start of the study. As we move out over time, we see that the number of patients still living diminishes. Finally, by the end of the study, we find that about 50 patients, or about 50% of the patients, remain alive. Now, if we compare Cohort A with Cohort B, we can see what is called a wide separation between the two curves. At all points in time throughout the entire study, more patients from Cohort A are alive than Cohort B. Of course, there are other variables which must be accounted for. But in the simplest interpretation, it implies that if you were choosing therapy blindly for all patients with a certain cancer type, you might be inclined to choose treatment A over treatment B. Unfortunately, it becomes a little more messy in the real world, where the picture is not as clear-cut as in this example. Very often, too often as we'll see in the next module, the curves are not widely separated. In fact, there may be no significant separation whatsoever. In this latter case, no conclusion can be drawn with respect to which cancer treatment might be superior. Unfortunately, this is what happens in most clinical trials of new cancer treatments. It explains why very little progress has been made in treating most types of cancer. It also clearly demonstrates why it is so important to stop conducting endless clinical trials, whose purpose is to identify the elusive one-size-fits-all cancer treatment, and instead to start personalizing chemotherapy for every cancer patient, as an individual, as we do here in our lab. Please join me for the next module of personalized cancer chemotherapy, where we'll look at Kaplan-Meier graphs from real-world clinical trials involving real-world patients.